Welcome to Biotech Stage. It's a stage where we meet with biotech professionals from the globe to discuss something very interesting, something very informative, and something very hot in the biotechnology field. Today, our guest is Dr. Ravish Patel. He got his PhD in biotechnology almost 10 years ago, and since then, he has been working in different biotech companies in India, including Cipla, Wockhardt, Sun Pharma, and currently he is employed as General Manager technical operations at Kudo Life Sciences. So welcome, Dr. Ravish Patel, on this stage.
Thank you very much for giving me this opportunity. And I would say this is my second podcast, and I'm really very exciting for the few round of questions.
Okay. Yeah. So, Ravish, I think there are a lot of things that we can discuss with you because you got experience in biosimilars and now you are working in cell and gene therapy. And both the fields, especially the biosimilars, have grown a lot in past 10 to 15 years. And cell and gene therapy is also now coming up very fast. So, if you could please share your professional journey, your experiences in this field, that would be great for our audience.
Yeah, sure. So I have started my journey with the Intas Biotech in 2007 back, and then I was deeply involved with the physicochemical characterization and biological characterization of the therapeutic proteins. So over the years, I had an opportunity to work across the diverse modalities from biotherapeutics to plasma-derived protein to vaccines. So this each offering unique scientific and analytical challenges. And during my tenure, I gained hands-on experience in impurity profiling using advanced analytical tools and learned the critical importance of the cell-based assays for assessing the product potency. So many times I have seen there are a lot of challenges in this field, mainly when I was with the entire biotech, and also I have inspired it. When this inspired this journey also inspired because I have also enrolled myself for the earn and learn policy which was there that time, and I have enrolled myself for the PhD program, which helped me to strengthen my scientific foundation and develop the alternative. So we have developed the orthogonal approach for the in vitro, so animal-based studies during those days when I was associated with Intas, and that is in vitro assays which use the cells without using the animal model. So one of the product which we used that time was the follicle stimulating hormone that was recombinantly originated, and
Mm-hmm.
that demonstrates the strong correlation with the traditional in vivo potency models. And so then onwards, if I talk about my journey from the 13 to 16, I played a key role in analytical method transfer across the globe at the client sites, which enhanced my understanding of the international regulatory requirement. And I have played a very key role in over a time. So I witnessed in the field of evolving from the biosimilar to next generation modalities like antibody drug conjugates, bispecific antibodies, immune checkpoint inhibitors, and the journey that naturally lead me to cell and gene therapy space. So this is how
Okay.
I would explain.
Thank you very much. And Ravish, we know that biotechnology, especially the biosimilars, have grown a lot in past 10, 15 years. And when the
Yeah.
industry is growing, obviously there are a lot of opportunities. But at the same time, there are a lot of challenges. I mean, opportunities does not come just as opportunity. There are some challenges associated with it. And people like you, professionals, they handle those challenges. They solve the problems. If you could please share some of the biggest challenges you faced, and especially, are those challenges you think are similar in cell and gene therapy industry? The challenges are different.
So now cell and gene therapies and the biosimilars are having a so it's a different ball game, I would say. So when we say that the scientific understandings on the biosimilarities is evolving since 2000 until date, it's keep on learning on the many gray areas and many things which we eventually learn from the.
Mm-hmm.
And I would say that the understanding on the biosimilars, we have a very great foundation, and that helps us to understand more about the cell and gene therapy space and this scientific rigor and comparability activities what we did during for the biosimilars. So these understandings help us to establish the critical quality attributes for complex molecules, which this help us to define the similar framework for the cell and gene therapy. Mainly
Mm-hmm.
when we talk about the critical raw materials such as plasmid DNA, lentivirus vectors. Understanding of this analytical this helped me to to understand the how analytical tools such as the advanced analytical tool and biological assays now directly inform the quality assessment strategies for cell and gene therapy raw materials. So regulatory alignment
Mhm.
also is really very important. So navigating the global biosimilar submission, which help us everybody currently all the manufacturer to strengthen the approach to the quality documentation, risk assessment. So all crucial for CGT regulatory compliance equally.
Okay. So it is good to know that biosimilar or the experience in the biosimilars or the development in the biosimilar industry made it basically a framework for the cell and gene therapy. But do you think the regulatory expectations, regulatory guidelines, they differ significantly between biosimilars and CGT, or they are quite different?
More or less they are similar, but yes. So this is really interesting aspect which I wanted to put a highlight on that. That the over the last 2 decades, the biosimilar industries has matured tremendously.
Mm-hmm.
And initially, the focus was on the establishing the analytical similarities and building the regulatory confidence. So today,
Mm-hmm.
biosimilars are an integral part of the global healthcare, supported by well-defined guidelines and advanced characterization platforms.
Mm-hmm. Mm-hmm.
Uh, so mainly in India, the journey has been particularly transformative, moving from the process imitation to true innovation and
Mm-hmm.
global standardization. So with Indian companies now gaining an international regulatory approvals and market presence. So this is also something which is backing a factor for the cell and gene therapy regulatory requirement. Yes, there are a lot of gray areas still there in the cell and gene therapy. We are
Mm-hmm.
still learning as we are growing in this field because things are very new, not only from the characterization aspects but also from the clinical studies, clinical trials, and from clone so cell to clinic and or cell to patient. So cell and gene therapies has a I would say that way to go ahead, and it's again a long journey, but yes, the
Mm-hmm.
platform has been set by biosimilar regulatory framework has tremendously helping to understand
Mm-hmm.
this space.
Okay, so again, it's great to know. As you mentioned, biosimilar industry has matured a lot in past 20 years, and
Yes,
that helped the cell and gene therapy to progress. So
yes,
that's
of
good.
course.
So Ravish, I also noticed that your company Kudo Life Sciences is big in plasmid DNA manufacturing. So and
Yeah.
plasmid DNA is used as a raw material in the biotech industry. So can you say something about, or can you discuss something about the plasmid DNA? I mean, how does it help the industry? Where it could be used, things like that.
Yeah, so plasmid DNA is the foundational raw material for many advanced therapeutic such as mRNA
Mm-hmm.
vaccine,
Mm-hmm.
also a gene editing platform like CRISPRs and immunotherapies, and also after the COVID, so the understandings is and then the requirement is also tremendously increased. So,
Mm-hmm.
and everybody knows that the how that situation has scared everyone globally. We were in a dire need of the vaccines. So and mRNA vaccines is booming like anything, so on. And now major pharma company, biotherapeutic companies are now focusing their side towards the not only focusing on biosimilars but also towards the mRNA kind platforms, CRISPR platform, gene editing platform, cell and gene therapy, and so on.
Mm-hmm.
So the quality, purity, and integrity of plasmid DNA is directly influence the efficacy and safety of downstream process. And establishing a well-defined critical quality attribute for plasmid DNA is therefore a crucial for ensuring the success and consistency of CGT product.
Oh, okay, okay, okay. So plasmid DNA, all other kinds of DNA that is used for
Yes,
manipulating the different.
yes, it's a one of the critical raw material, I would say.
Okay, okay, and obviously it is grown in bacterial cells.
Yes, yes, yes.
Can you say anything about the like the scale? How much scale, or what is the largest scale you guys produce, or it varies from order to order?
Yeah, so it varies from order to order. So currently at the Kudo Life Sciences, we take a pride in being the first Indian startup to
Mm-hmm.
deploy our single reactor for plasmid DNA manufacturing. And this innovation not only enhance the process flexibility and scalability, but also reduce the contamination risk and turnaround time.
Okay.
So that is
Okay.
what I would say that it's healthy using the SUF.
Okay. 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 And how long does it take from start to finish, growing the very first batch of bacterial cells to a purified lot of plasmid? Does it take few hours or few days?
It's the total 4 days process.
Okay.
It's not a 2 hours process. So total from end to end. So it takes 4 days. So upstream, downstream, and
Okay.
we usually run our bioreactor for almost around 18 to 20 hours for the upstream
Mm-hmm.
part.
Okay. 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 So Ravish, so you are working as a general manager for technical operations. So when it comes to technical operations, there are a lot of responsibilities, and I'm sure at the startup company, and you just mentioned Kodo is a startup company. People are more busy at the startup companies because you need to wear different types of hats. You are a technical manager. You are doing also a lot of other responsibilities. So can you just share your experience on a day-to-day basis? What are your responsibilities on a day-to-day basis?
So as a general manager, technical operation, I oversee end-to-end manufacturing strategy, analytical development, and regulatory element. Sometimes I involve in that also. Mainly, I'm looking quality control aspects. So quality control, uh, which is very important and which is considered as a heart of any manufacturing unit, and
Mm-hmm.
also quality management systems which involves that. So working in a startup environment means wearing a multiple hats.
Mm-hmm. Mm-hmm.
So driving the process innovation, ensuring the GMP compliance, monitoring the young scientists, and compared to the large pharma, the startup ecosystems offering eligibility, faster decision making, and culture of an
Mm-hmm.
scientific entrepreneur. So this
Mm-hmm.
is what actually the opportunity is in this role, I would say. And yes, of course, I got in also opportunities to share my knowledge on the international platform since in and recently, I also participate in the one of the very touted conference that festivals of biologics in the last month. So there also we have gotten lot of opportunity to interact with the different part of the world who came there as a speaker, as a delegates, and very few fine scientists also. So in
Mm-hmm.
this space, I got a good opportunity to understand that what are the challenges when we compare with the USA versus the European countries. So,
Mm-hmm.
and each time I attend such a conference, I got a lot of understanding on this part and get a very different flavor. I would say that how this tremendously evolving, and I'm sure the day is not so far. These new modalities and precision medicine would be completely change the the treatment for rare diseases, I would say.
Mm-hmm.
Uh, there is the only challenge which I would currently see is that the economics part, which is something which is really, I would say that is the major road blocker. I would use the word, and that is something uh, collaboratively we can work, and then we can reduce the overall cost to the patient.
Okay. Okay. So, Ravish, you have worked in companies like Cipla, Wockhardt, Sun Pharma, which are the big pharmaceutical companies. And Kudo Life Science is obviously much smaller than that. So, can you share anything on like how the working at a small startup company is different than large pharmaceutical companies?
Yeah, so Indian startups have a unique opportunity to bridge the affordability with the innovations. So that is how
Mm-hmm.
I can relate with this. So as you mentioned, that I have worked with all branded company also, and now with Kudo Life Sciences, which is startup, but no longer will be startup. And
Mm-hmm.
Kudo
Mm-hmm.
is also someday would become definitely one of the branded one, like the and this is what the we have started our journey with the Codo Life Sciences in keeping in a mindset for the affordability. And the challenge
Mm-hmm.
lies in regulatory harmonization. So that's what something which we need to look into. And that is what I have also understand during my past few conferences, that we are discussing on this part and on very high note, that regulatory harmonization, talent
Mm-hmm.
development, infra. So these are the few things which I have eventually understand. And also we are also lagging a lot back when we are not working hand in hand with the academics. So as I have ended up with the Kudo Life Sciences current, the potential is immense. With the right ecosystem, India can emerge as a global hub for CGT manufacturing, just as
Mm-hmm.
it did for the bios. So the motto of this, to be part of this new venture, is bring to the value to the global space, and that is what actually we as a team are trying.
Okay, okay. It's great to know that small world startup companies, they offer a lot of innovation, and they are a startup, but tomorrow they will be a big biopharmaceutical company.
Yeah,
Yeah,
of course, and that day is
yeah.
not so.
And it is all the hard work of our different employees and the management. So that's great to know.
Yeah, yes,
Mm.
and we are committed and we are determined to do.
Yeah, yeah. I mean, I can tell by the excitement you have and all the nice stories that I hear about different companies on the LinkedIn and the social media. Yeah, that's great to know. So I also wanted to ask. So these are different opportunities that company can grow faster and company can become the large pharmaceutical. But what are the challenges that a startup company would face, especially in cell and gene therapy area?
Uh, so we are yet to have that maturity in this field. So I would say mainly from the regulatory, that is the one. Second thing, which what I foresee is that what are the quality attributes for this
Hmm.
kind of. The third one is
Hmm.
the efficacy assays, which is I would say the potency assays, and
Hmm.
what is the you know I would say right right approach, to demonstrate the potential, right approach to demonstrate the efficacy of the CGT products, and also how we can correlate with the clinical relevance. So this is all makes a very big and unique things when we talk about the submission to the regulatory bodies. And there also people is very open to offering, very open to, you know for the discussion, I would say, before you are submitting any regulatory filing. So
Hmm.
things are also now changing, and the analytical discipline and regulatory experience from the biosimilar definitely help to accelerate the CGT maturity. So in many ways, the biosimilars laid the foundation for the next wave for the advanced therapies. In my opinion.
Okay, okay. So in terms of growth, do you think these two, biosimilar and CGT, they are similar or they are going to be fundamentally two different worlds?
No, no. What I sense and I experience since joining Kudo Life Sciences, since I have worked 18 years in the industry, I see that this is a very, very similar when we talk about upstream part, when we talk about the downstream part, when we talk about regulatory per se, when we talk about the quality control per se, and analytical aspects, characterization aspects, very, very similar, very, very similar techniques.
Mm-hmm.
And there are definitely compoundal methods which is completely replicated here also similar. So
Mm-hmm.
I don't there is a much more differences between these two space. They are morely similar, and and also seeing, if you see that the flocks we hired for this, they are not
Mm-hmm.
experienced in the cell therapy. They are all biosimilar guys, and
Okay.
currently the team also, you see in any organization who are currently working on the CGT space, they are something not they have learned anything because this field is also new. So you will be whatever the experienced scientist, they are also learning eventually with their
Mm-hmm.
background and their understanding on biosimilars.
Okay. And Ravish, now we are coming towards the end. One thing I wanted to tell you that Target audience for this podcast is a younger generation. Those people are hungry for information. They want to learn. I mean, they got the degree, they got some experience, but they are trying to find out what they can do to improve their growth, what they can do to go to the next level. So can you share or any advice you want to give to the young scientists who are entering into the field or have a few years of experience so that they can excel?
Yeah, so my advice to young generation is currently we have a lot of blogs which are joining which have already joined the Kudo Life Sciences. They are very enthusiastic. They are very they have a lot of ambitions in this field where they wanted to learn, they wanted to understand a lot of new things. I wanted to give one very nice piece of advice is don't let your enthusiasm down if you see any failures, because the failures
Mhm,
is something which is first step of the success. So this is what actually I have also learned. And uh, morning only I have an opportunity to speak one of my colleague. There was an instance in today's activity where we were analyzing some data on our ongoing batch, and we observed some setbacks. So what I understood is that people who are the young scientists, especially young scientists, they are very hesitant to to look into the orthogonal aspects, look into the other angle, and mainly look out of the box.
mhm,
So I have interacted with my colleague, and I explained him that believe me that always the first step is always very difficult, and when
mhm,
you climb that first step, the journey is really not that difficult. Obviously, you
mhm,
might be feeling some setbacks, some obstacles, and there might be
mhm,
some hiccups. This is also a part of the journey, but first step is always difficult. So don't never ever get hesitate to take that first step.
mhm.
So
Yeah.
that is my advice to the young generation, young scientists. And how science translate to the patients is mainly impacting and be adaptable, because the
Mhm.
biotherapeutics world is evolving faster than ever.
Yeah, and that means a lot of growth opportunities. I think your advice is very good. I mean, I never ever hesitate to take the first step, because if you don't take the first step, there won't be any second step. So thank you very much for the advice.
And this is really very important.
So now we are coming to the end of this podcast, and we are going to end this with a rapid fire question segment. So I'm going to ask you 10 questions, and please give your response from a scale of 1 to 10, 10 being the best or almost there or strongly agree depending upon the question. So, and we will try to finish this in less than 60 seconds.
Yeah.
The very first question is quality of biotech drug products in India.
8,
Okay. Production capacity in India.
7,
Okay. Innovation in India.
4,
Okay. And IP protection in India.
6,
Six. Adequate availability of talented workforce in India.
9,
Okay. Productive interaction among peers.
7,
Okay. And capital available to support new projects.
7,
Okay. Cost efficiency with an aim to cut down the cost of final drug.
7,
Okay. And unambiguous and clear guidelines for regulatory purposes.
7,
Okay. And finally, make in India efforts.
10.
That's 10. Okay. Okay. So with this, we finish the rapid fire question segment. And also, this is the end of the podcast. So thank you, Dr. Patel, for sharing your experience and giving the guidance to the new generation and discussing all the challenges and opportunities in the biotech field.
Thank you, Dr. Arun, for giving me this opportunity to join your podcast as Biotech Stage, which is a very souted and prestigious biotech stage. So it's now literally people are getting a lot of understandings about your new this venture that giving a lot of understandings, insights on the biotherapeutic molecules, vaccines, and cell and gene therapy. So really amazing time altogether. Thank you very much once again.
Yeah. Thank you.